VCR. Okay. Okay, folks, welcome. Let's see. Turn this down a bit. Testing, testing. Okay. Hello. We haven't seen each other for a week, so we're all going to have a lovely commute after class today, I'm sure. Okay. All right, so I doubt there may be one or two of you, but if you just added the course and you don't have access to Blackboard, you can just email me, but you should have access by tomorrow or the next day. But I can at least send you some material, okay? If you see Blackboard, then you are good. Everything is there that you need, okay? Of course, if you have questions, just let me know, okay? So, so we are doing clickers for real today and throughout, of course, hereafter. And so I figured we just kind of ease into this with a relevant question, relevant in the world of what happened over the weekend. And so just kind of we have another 2 weeks yet, a week and a half. So let's kind of gauge the pulse of the nation, so to speak. I really don't care at all, okay? And like, if you were like yesterday's class, you really didn't care. You really didn't care. So, so let's see what we come up with here, okay? So the Super Bowl edition question, okay? So kind of, again, just to kind of break into the clicker questions, right? So the winner will be whoever you think, okay? Yeah. Yeah. Okay. So timer's on. Okay. Okay. Okay, so we got like 210. So if you're a straggler, put something in. Okay. Most of these choices are for those that maybe darn aware of who's in the Super Bowl, or think that J.J. Watt is, will win it. Okay. So, okay, actually, he's I think he's hosting Saturday Night Live this Saturday. So, I heard that go, oh my God. Okay. So, all right. So the majority yesterday said, who cares? It was like 34%, I think, was the total. Between DC and BIOL, yeah, okay, who cares? JJ Watt got 9%. That's pretty good. I've always got 4%. Tom Brady 3%. We got more. Okay. I guess he appeals more to like maybe the women in their 40s and 30s and 40s, maybe. I don't know. Anyway, so maybe not. Okay. All right. So I really don't care either. Of course, I'll get rocks thrown at me if I tell you who I'm for, because it's on there. I'm a Cowboys fan. So you can go ahead and boo. But that goes way back. We're talking the Super Bowl V, right? Since then, those days. So, and it was Super Bowl before, way back then. Okay, those days don't know that. So anyway, so let's do. What we're going to do is we're going to get through chapter. Finish chapter off. I doubt we'll get into much chapter 3 stuff, but hopefully get through all chapter 1. And so we're going to have another, maybe call it more legitimate, quicker question here in a couple slides. So, just be ready after this slide, actually. So, this is more just kind of a recap, right? So, what have we been talking? This is basically chapter 1 is here's what microbes are all about. Here's how we define them. Kind of the who, what, why, when, and where. Here's a good, if you want a good, test. Do you know chapter 1 stuff? How well do you know it? Put who, what, why, when, and where on a sheet of paper, and see how you can fill that out and define. And so see how much you can fill that out. So we kind of started with defining microbes, and we went to kind of discovery, Van Leeuwenhoek, etc. Then kind of into the spontaneous generation. How do these things come about? Is it just through an abiotic method of just bringing forth from non-living things? Of course, that's not true. So we went into Pasteur and what he figured out with germ theory of fermentation, that these chemical transformations that occur, many of these in nature, of course, can be attributed to living things doing these chemical transformations. Metabolism, right? And so, and then kind of linking that to germ theory of disease, only in the context of microbes are transforming organic materials to end products. A infectious disease state could be transforming this organic body into not necessarily end products, but into a disease state. And so, of course, Koch kind of laid the groundwork for that. And then we looked at some kind of the exceptions to Koch's postulates, and then kind of a little bit about working with microbes, culturing, etc. And I think we ended on the slide with the basis of immunity. Okay, so we looked at looking at that many microbes can cause disease. Then how do we get rid of them? Right, through vaccination, chemical means, etc. Okay, I think that's where we ended last time. So let's. So this next question is, there's one of the things I do with these clicker questions is I'll have the same exact question and show it twice during the lecture. Okay, we'll throw it at you first, then you'll have we'll go through some material, then we'll throw it up again, and then see if we see. And this is one of those, right? On this slide, there's 3 areas. 3 of these statements we actually did touch on last time. The rest of them we did not. Okay, so kind of stuff we're covering today. So please, channel 41. Channel 41. Okay, on your clickers. So 3 statement, see what you think, and then when we're done, I will touch on 3 of the ones, and then I'll save the rest for the end of class. Okay, because we'll see this question again. Okay. And remember, if you choose to do so, you can confer with your neighbor, right? If you're not sure, right? Or neighbors, plural. Okay. Okay, timer's on, so think about it. All right, 1 second, speak now, and here we go. Okay, so 41% A, that's very interesting. Okay, so that brings up a good point, but what I'm going to do is touch on these three, because we talked about those touched on those last time. So, so using a live non-attenuated vaccine is a safer choice than using a live attenuated vaccine. So remember the key word attenuated, right? Attenuation means to limit to essentially in this context is to using a vaccine that's not using the full-blown disease-causing form of the pathogen, right? Whether it's virus or bacteria, you are chemically neutralizing it so that we keep active the parts of that pathogen that will activate the immune system, right? Those antigens, okay? We attenuate it to that's what we call that process, to inactivate it. And a non-attenuated vaccine, of course, is the full-blown pathogen, and of course that can you can cause disease if you get that as a vaccine. Pure culture isolation, so you can't use only liquid media, okay? Solid media has to be part of that, okay? Because you need to be able to, you can't manipulate individual strains in a liquid suspension, right? You need to have a visual of them on a plate, right? Solid media that you can then have them grow as colonies on the plate. Then you can see, oh, this guy's different, this one's different. Let's pick those and then do something with them, culture them again, right? Subculture. So liquid media won't allow you to do that, okay? There's a role for liquid media we'll talk about later, but not, and it can be part of the process, but it can't be the way you ultimately obtain a pure culture, okay? Koch's postulate says that for a specific infectious disease, healthy individuals can possess the pathogen. He did not think that, right? He thought only diseased animals / individuals would carry a pathogen, not healthy people. He wasn't aware of that could be the case. And so that's one of those kind of, it doesn't always work. Koch's postulates all of them don't necessarily always work because we have these other things that can occur, and that's the carrier is one of those, okay? So B, D, and G is written are all false here, right? So we'll touch on A, C, E, and F as we go through, okay? So, so we talked about vaccination. So antiseptics, disinfectants, okay? So using chemical agents to control microbes in terms of their growth, temperature, pH, however you are trying to control the growth of pathogens, all those most methods are meant to reduce their numbers. Reduce, not eliminate, okay? People are very confused about what the term sterilization means. Now, when I talk about sterilization, I'm not talking about it in the context of you can have kids, right? Not that kind of sterilization. Talking about sterilization in microbiology is if you have sterilized something, okay? If I have sterilized this square foot of bench, nothing exists there, zero. There's no life in that area, okay? No viruses, no cells, nothing, no life, dead. Okay, that's what the sterilization method does. And antisepsis doesn't do that. Disinfection doesn't do that, okay? They reduce the numbers of pathogens greatly, okay? That's what you do in lab. When you're spraying your desk down with ethanol, you're not sterilizing, okay, you are reducing numbers significantly, okay. You'll see on the bottle of Lysol, kills 99.999% of bacteria, viruses, what have you, okay. It doesn't mean it's going to zero. 99.99% is not going to zero. It could be close, right? Because it depends on how much contamination you have, right? If you have 1 million cells, you still got a significant number of cells left, even after an agent that kills that much. So, point is, autoclave can sterilize. Okay, there are certain agents, radiation can sterilize. Okay, so just be wary of how you use that term sterilization. Okay, so antiseptics and disinfectants do just that. They reduce the levels of pathogens. That's what they target. They target pathogens. Okay. And so, Semmelweis and Lister. So, the basic difference between an antiseptic and a disinfectant is an antiseptic you can use on what? Humans on living tissue. Okay? So, isopropyl alcohol, right? Bleach, no. Right? You wouldn't want to wash your hands with bleach. So, those are for inanimate objects, a bench top, surgical instruments, right? And so, doing these kind of what we nowadays think of as really basic things to disinfect or use antisepsis significantly reduced the instances of infections. So Semmelweis was a doctor working in a hospital, and he found that in his hospital where he worked, that the cases of what's called childbed fever among women are super high compared to women who would not come to a hospital to have a baby, but would have a midwife take care of it, okay, outside the hospital in their home or what have you. Much less infection, much less cases of childbed fever. And it turned out that the doctors in the hospital were just walking around like slobs, right? Just fluids and blood on their lab coat, on their coat, not really taking much care in terms of washing hands between patients and whatnot. And so it's no wonder that they had such high levels of infection. And so simply just washing hands using things like lye soap and whatnot would really reduce the incidence of disease. Lister did the same thing, but more so with somewhat with antiseptics, but also with things like surgical instruments and things, disinfecting those and disinfecting the area where surgeries would happen, okay, in the rooms and whatnot. Significantly reduced infection. So antibiotics, of course, Fleming and the discovery of the mold that produced penicillin was, obviously worldwide impact, right, in reducing disease. But of course, microbes, like other living things, evolve, right? And there will be types that will be resistant, of course, and we all know this, okay? So one of the things to remember back from intro bio when learning about evolution is that resistance depends on the presence of the antibiotic, right? That's the selective pressure, right? So if that's there, then if there are members in a population, right, that are resistant, then they're the ones that will survive and reproduce and permeate throughout the population, okay? So the point is that there already will be mutant types in the population that, when exposed to an antibiotic, it may enable them to survive. The mutation may be enough that changes something that they can survive, and then they're the ones that everybody else is wiped out, but they reproduce. Their numbers increase. That's how resistance occurs, okay? And nowadays, and for some years now, there's antibiotics everywhere. Yeah, you go to the doctor's office, you have an infection, you get an antibiotic. Oftentimes, it's what we call a broad spectrum antibiotic, okay? The doctor doesn't really want to, likely doesn't have the time to isolate what you've got. So they say, well, take this, it'll cover a range of bacteria, okay? People go to the doctor thinking they have a bacterial infection, and they don't. They have a viral infection, but they still get antibiotics. So they're still exposing themselves to antibiotics when it's not going to do anything. And then you take it out of the doctor's office, and now environmentally, Everything we eat almost has antibiotics. Chickens, the chickens you eat, the cows we eat consume, full of antibiotics. Okay, milk, right? A chicken breast is not supposed to be this big. That's not natural. Those are hormones. Okay, and then you take wastewater samples, and you'll find antibiotics in there as well. So these are all opportunities, right? They're out there. Bacterial populations come into contact. Those that are resistant, they begin to flourish. This is how this spreads. And now we're dealing with bacteria that are multi-drug resistant. And so I'm going to show you this, but don't worry about this. It's only for just to show you kind of thing. Okay. We'll talk about more about this later in the semester. But in terms of resistance, there's kind of, 3 common ways this happens. Okay. So here's an example of here's an antibiotic. Here's a bacterium. So antibiotic comes into the cell. One of the things that can happen is an existing transport protein the bacteria has can be, it may acquire a mutation that changes this slightly so that it's specific for pumping the antibiotic out. What's called efflux. Efflux means to go out. Okay, so that's one mechanism. Another one is an antibiotic will have a target within that cell. Okay, it could be part of the protein synthesis machinery or some other target. And so there's going to be a specificity in binding antibiotic to target. Okay, and so it may be that the bacteria acquires a mutation that alters the shape of that target. So now it can no longer bind, and so it's not affected by antibiotic. It could simply also be that it has an enzyme that basically just chops up the antibiotic as it comes in. That's what penicillin resistance, one of those is an enzyme called beta-lactamase that basically chews up the penicillin. So all types are fairly common resistant types. And again, they can be multiples of these in cells. So it's obviously a real issue. And the pre-pharmacy people always have something to work on, trying to figure out, coming up with new drugs and so forth to target these things. So, okay. Any questions? Okay. So let's look at ecology. There's a couple of slides in here. I've kind of changed the order. So I forgot where I'm at. So if they come up, I may hesitate for a second. But I think there was a reason for me doing that. But anyway, so microbial ecology. Okay. So the discovery of different metabolic types. Okay. Up to this point, through Pasteur's work and others, we learned of transformation of organic materials, right? How we eat, right? How we eat stuff, right? We take organic materials, break it down, get energy. So they found bacterial types with these unique metabolisms, okay, in terrestrial environments, soil, aquatic environments, and sediments. And they would take these samples, whether it was mud or other soil-type samples, and they would expose it to various substrates, and they would see that these substrates would be transformed by agents in the soil. Okay, and so, for example, hydrogen gas could to the soil sample, and they would see it being transformed, going away and transformed into other inorganic compounds. They could expose it to ammonia, and ammonia would be oxidized to nitrate. So they attributed this to microbial activity. What's kind of a soil sample right here? All right, 2 soil samples. You expose both to, let's say, ammonia. Okay. If you wanted to prove it was biological activity that was doing this, what could you do? So one sample, it would be your treatment, right? What would your control be? How could you figure that out? Any idea, right? If you suspect the material in one is causing this, you might not expect to see the activity if you did something to this one. What could you do? Yeah. Because you'd want to Right. You want to, right. You autoclave it, some kind of chemical, some kind of treatment to basically kill what's in there. Okay, and then you could hypothesize, okay, well, I've destroyed whatever might be a living thing in here because I did X treatment, so I shouldn't see any uptake of hydrogen or ammonia, what have you. I would see it in the one where I didn't do that treatment. And so that would be an indication that, yeah, that's what's going on. That's what they did. It turned out that these kind of organisms can be very hard to cultivate, especially with the technologies of the 1890s, 1880s. We have since been able to do it, but they still don't grow super great, but we can at least nowadays. But we're talking about our lithotrophs. So they discovered lithotrophs. And so these types of bacteria can use completely inorganic materials, okay, as an energy source. Like we use glucose and can get energy from that. They use things like hydrogen gas, H2S, ammonia, even iron, 2+, right? And so, so this activity is very important in terms of ecology. And this Winograd column is what they developed to study these guys. And so basically you fill up a jug with like and I've done this once. It takes a long time to finally see something like that. But to the technology center, there's like a pond over there. Took some of the shoreline, took some of the kind of the mud and plopped it to mixed it up with I didn't add these chemicals off the shelf. What you can do is you add the newspaper, you add like eggshells. Eggshells are calcium and sulfate and even calcium carbonate. So you can add that as eggshells to keep it pretty kind of basic. And then kind of put it on the shelf, expose the sun, just let it sit. I mean, that's all you do, just let it sit, okay? So what you see is the activities of different and come forth, okay? So the newspaper in this scenario has what purpose? Microbes reading the newspaper while they're in the carbon source. So that's the carbon. And the carbon in the newspaper is primarily, cellulose. So so it's an organic source of carbon. So you'll have what are called trophic types like us. Okay? They'll begin to chew on that, and they'll basically get rid of any residual oxygen in there that may be in there. And then it starts to go anaerobic, okay, as the oxygen is taken away. And then you begin to get things like fermentations, right? Metabolism without air. And so fermentations produce gases and things. So then you may get hydrogen gas. And now, when H2 forms, then you can, that's what your lithotrophs like to eat. Those can eat, okay? And so they may begin to use that. You'll have types that can reduce sulfate to things like H2S, okay? And now you have some that kind of guys can use. They like purple sulfur bacteria, green sulfur bacteria. They like things like H2S and light, and they can photosynthesize. So they discover photosynthetic types that are very different from cyanobacteria. So remember, these guys are bacteria, but they photosynthesize like plant, like algae, okay, using water and giving off oxygen. These guys do not. So they use things like H2S, and iron in the place of water, and use that to photosynthesize. So you'll see different layers, okay, and they produce different layers because these guys use different wavelengths than cyanobacteria, and they actually use lower energy forms of light. They get to be they stratify below the cyanobacteria, which is producing oxygen up here. Then you have the sulfate-reducing types, which are anaerobic. So you get different types of lithotrophs, synthetic types, and even some various colors. Here's like a purplish and a yellowish color, kind of to their photosynthetic pigments. Okay, so so you're seeing then what's unique about these lithotrophs is this. So their activity forms of nitrogen and sulfur, phosphorus, that all living things have to have, okay, I'll show you in context. So two nutritional types, right? So remember this, because we're going to see this certainly in chapter 4 on growth, but it's one of those basic things everybody should know, what a heterotroph is, what an autotroph is, okay? So basically, if you forget, you're a heterotroph, okay? You use complex organic compounds. You define these two types by the carbon source. That's how you define it, okay? So an autotroph uses CO2, okay? So your chemoautotrophs, photoautotrophs, and truthfully, a chemoautotroph, okay, is the same thing as a lithotroph, okay? So this is the same as that, okay? So lithotrophs can use inorganic materials for energy, right? We can use glucose, right? C6H12O6, right? We can break that down, okay? So we can oxidize this, get energy, right? ATPs, right? But it also has another purpose, right? It has this, C, carbon, right? Got it? We're carbon-based life here on Earth, right? All our molecules are built around a carbon skeleton, right? And we have things like nitrogen and phosphorus to make nucleotides. You can add nitrogen to make and phosphorus to make amino acids. Okay. Molecules are constructed around that carbon skeleton, right? And adding different atoms to it to make different variations, right? Of carbohydrates, lipids, DNA, RNA, etc. Right? So now a lithotroph uses an inorganic form of energy, ammonia, H2S, right? So that's whereas we are dual purpose, we can get both carbon and energy from a single source. They cannot, okay? So they have an energy source, say ammonium, that they can make ATPs with by capturing energy by oxidizing this. But what about carbon, right? That's why they're autotrophs, okay? Fixed CO2, right? So many of these are types of examples of because I'm assuming that what you learned to this point is the only type of autotrophs in your heads are plants in algae, right? But there's a lot of types that do not need to have, right, as the equation, right? They get the energy from these inorganic types and then use energy CO2, okay? So that's your lithotrophs. And so their activity, of course, is essential. And so we look at where do heterotrophs, autotrophs fit in, right? So this is looking at our, from ecology, kind of the feeding levels, right, in an ecosystem, that your producers are at the bottom. Autotrophs are at different levels. And so any ecosystem, the foundation is going to be your autotrophs, right? Your plants, algae, etc. Okay, and, they of course your heterotrophs give off CO2. Okay, that's what producers, autotrophs, use, fixed CO2. Okay, and many of your producers will produce oxygen, right? So through the photosynthetic actions. And so not all, but most do, and at all levels, so decomposers are also very important. So at all levels, right, bacteria and fungi, decomposition, right, microbes are at all levels, okay? They're heterotrophs, they're types that are producers, okay, at all levels. And so decomposition is particularly important because here and here, in these two groups, is going to, right? That material can be broken down by decomposers, and then now you're releasing nutrients that others in the ecosystem can use. Trophy release forms that other in the ecosystem can use, all together. Okay? And so one of the important cycles in ecosystems is this one, the nitrogen cycle, or triangle, as it's called. And all sides of this are driven by different bacterial types. Okay, it's pretty much a totally bacterial prokaryote process on all sides. Okay? And one of the important things that does is to take N2 from the atmosphere and fix it, right? The fixation reaction of nitrogen gas to produce ammonium ion. Okay, that's huge. You know, the amount of nitrogen here, 70-something%, right? So you have you're bringing that into an ecosystem. Now we have nitrogen being brought in to an ecosystem where it can be used. It can't be used out here in the atmosphere. So material types, the most important ones of this activity have associations with plants. Okay, and it's how we get nitrogen into those ecosystems. Okay, both aquatic and terrestrial ecosystems. Okay, this process of nitrification, this is the actual lithotrophy. Okay, so that is where we're taking inorganic source and convert it to another inorganic form, nitrite, and then nitrate. Okay, these forms like readily assimilate that. So even though the producer level, plants and algae, they don't need a lot, right? Just give them some sunlight, some water, and CO2, which they get from the air, and they're good to go, except they cannot make their own nitrogen or phosphorus, and so they depend on this activity to provide that to them, whether it's aquatic systems or terrestrial systems. So that microbial activity is very important. And so you have to remember, right? Because when we think about where are humans in this process, right? Because oftentimes we kind of separate ourselves from this, or we don't associate with these types, right? We're our own superior beings, right? So, but remember that obviously we're in this group here, okay? But we rely on these guys, okay? And these guys rely on this activity, okay? So ultimately that means, logically, that we depend on these as well, okay? Because without forming this, you aren't happy, your producers aren't happy, then your different levels of consumers aren't happy, right? So all goes together, right? Which is why I say, without this activity of these various prokaryotes and keeping that, maintaining it, we are doomed otherwise, okay? So, all right. Any questions about that? Right?
Well, how physically complicated is a fixation? Otherwise, we could use like plants never evolved the ability to do it itself.
Well, number one, probably because plants didn't evolve it to do it themselves, is this process of going from N2 and fixing it is a super energy requiring process. It takes a lot of energy to do that, and a number of different things, like 20-something involved, but very, very energy expensive, and also very oxygen sensitive. Okay, so bacteria that do this actually have little compartments on the plant roots to kind of help keep it anaerobic. And actually, they have the bacteria that do this actually produce a type of analogous to hemoglobin. So the role of it is to bind oxygen so that it doesn't poison that reaction. So for a plant to have it by itself probably wouldn't have been favorable, because it would be constricted in terms of sapping a lot of energy to do it. So it's more efficient if it has something else, make an association with something else and have it do it and exploit that way. Okay. Any other questions? Okay. All right. So when we get to metabolism again, just commit this to memory if you have to. This, and this is probably a good idea too. Okay. Because all right. So now I'm sure it's obvious to you that we talk about pure culture and then in a lab and doing things aseptically, and contamination, etc. Obviously, that's a fantasy in terms of how it really is out in the environment. Okay, nothing is in the pure culture out in the environment. So we have this is just kind of okay. I'm sorry. So this is meant to show kind of in nature, bacteria are going to in association, obviously, with bazillions of other microbes. Okay, and for many of them, they will have relationships where the product of one metabolism feeds another species. Okay, and so these feeding relationships they call syntrophy. Syn means together. Trophy, eat, so kind of eating together. So you may have organic compounds here converted to various types, certain types. They make things like and that may support the growth of lithotropes in the area. So they're all kind of one big happy family, if you will. Okay, and there can even be molecules between them each other, maintain these associations, sharing molecules perhaps. But this is reflective of what's reality in nature. And it's one of the reasons, probably one the biggest reason why we have of the microbes we've isolated, brought into the lab and studied, numbers around 4,000, I think. There are way more out there we've never been able to grow in the lab, right? Because we have not figured out these kinds of relationships, right? We may not be able to grow this one in lab because we don't know what are the nutrients it's using from something else to grow, right? That could very well be why we can't. So most that are out there are out there. We haven't yet taken them in to isolate them, right? There's still the bacterium that causes syphilis. We've been aware of that for 120 years, okay? Grow it in the lab. But fortunately, we can identify it through microscopy. If somebody has syphilis, we can identify it through microscopy and immunological tests, right? But can't culture it, even to this day, okay? So, and that's true for lots of different organisms. Okay? But so the something to remember. So endosymbiosis. So when a symbiotic relationship, I think we all know what that is. That's when you have two members have a relationship where they can be of different types, right? It may be beneficial to both, maybe beneficial to one but no effect to the other, or maybe one. So things like predation, when it's an endosymbiosis, that's an intimate connection between the two. One is either on or in the other one, and that's how the relationship is about. Okay, and so certainly the bugs that we have on us and in us, right? There are 10 times more of them are on in the body than our cells. Okay, and so the things that they've been around obviously as long as humans have been around. We've been carrying these things on in us for millions of years. Okay, and it's not just by random that their microbiome. So every day you find that there's something new about what they do for us. Okay, everything from enhancing our immune system, to allowing us to digest certain foods we couldn't, or certain molecules we wouldn't otherwise. Everything from controlling certain processes. There's a link between your microbiome and obesity, for example. Okay, even it's thought maybe autism between micro. So it's a different thing, and it's not surprising. Been carrying these things around with us for millions of years. There has to be something there. It's not just random chance. Okay. Yeah. We just talked about this. The plant important. I'm not just listing every type of endosymbiosis, but just some of the more prominent ones. Certainly with ruminants, right? Cows, other livestock that have multiple digestive chambers, right? It's when a cow grows up from a calf, right? And from eating grass and grain and whatnot, it's expanding really due to the activity of microbes in its gut. And it can be differentiated into different microbes in different that are doing different things. And the bottom line is to convert that material, the grass and grain, into nutrients the cow actually uses and absorbs. Okay, so it's all driven by different types of bacteria in the system that are doing that. Okay. Now, so here I threw this in just to illustrate the next point. And so if we have an environmental sample, transfer to solid medium, okay, if it's, we'll certainly see some different types show up, okay, as indicated by two different colony types. So here's this yellow guy. There's kind of maybe this cream-colored one here. That is two different colony types, and they're going to be two different species of bacteria. That's not always the case. You can have two different species that may have very similar colonies. So don't think that because they're just two colony types are the same, that they're going to be the same organism. It depends on the sample and what you've done, but just remember that. Regardless, so with all microbes in the sample here, say you took a handful of dirt, thrown it in, incubate and spin around on a plate. Is everything represented on that plate? Yes or no? No, of course not. Because you're not going to know what all the variables are in there. So, of course, you're going to represent a fraction. Okay? So the how, we all know, so now we have pure cultures, right? Okay. Now, the point is, you can do, and of course with these cultures, do whatever you want to do with them, okay? But the point back here, what about those types that didn't grow? Right? What if we try different conditions and different media, right? We still get nothing to grow, okay? What can we do? We have been able to grow through metagenomics. So that's how we can figure out what's out there in the environment without having to culture them. Okay, so basically what we're doing is genomes, right? Comparing genomes, taxonomic kind of approach. Okay, so we take the same kind of sample. We're just going to extract all the DNA. So you're going to extract the DNAs of all the things that are in there. Okay? And so you'll then chop up the DNA. Okay? And so using kind of the basic recombinant DNA / cloning techniques, you'll end up with DNA fragments of all the various living things in that sample will be cloned into vectors and then into, let's say, an cell. The purpose of the E. coli cell is to really just to grow up your DNA containing your fragment. That's the sole purpose of it. Okay? So what you have is a bank of cells, now a library of cells, where each one contains a of DNA that you isolated from the environmental sample. Okay? So you can be representing hundreds, if not thousands, of organisms in that sample. And you can do different things with it. You can go, okay, well, look at the sequence. What's the DNA sequence? Okay? So if you do that, you can go online. There are databases. We've been sequencing bacteria and everything else for 40 plus years. Okay? So there's a huge database of information. Okay? So you just basically just putting your computer to work. Okay? So you take your sequence, right, and you put it in there and see what's it similar to, right? Then you can begin to kind of, oh yeah, it belongs in this group. Or maybe it's unique, or maybe it's, that's how you can begin to figure out what you've got without having to culture it. Okay? You can also use that bank of cells and go, okay, I don't care what they are and what the sequence is. Maybe you want to make some dough, all right, and exploit what's in it. This kind of super whiz-bang enzyme activity, right? That will be the next, $1 million idea, okay? Where it can transform some kind of enzyme, right? So you can scan that library for the proteins, the genes that are on those fragments, and see what you get, right? Two approaches, but bottom line is this is how you can figure out what various microbes are in the environment without having to culture. It's the metagenomics thing, okay? Metagenomics technique. This is common nowadays on how you do microbial ecology and figure out, okay, what kind of bacteria are out there and how they're interacting and this and that. Be impossible to do if you had to culture everything, because you wouldn't be able to culture everything. Okay. Any questions about that? Okay. So, okay, I threw this one just for fun, right? I just want to get the old brain, thinking about beer, get the old brain gears going here. So just think about it, okay? So we have a gross medium formulation, okay, shown here. What type of microbe utilize all the nutrients listed and exhibit growth? Okay, so don't add anything to this, all right? Test 101, all right? Don't overthink and add, well, I thought this was added to me. No. Just with what's on here, okay? What would grow on this, okay? So there's another thing you need to remember is the it's not quite an acronym, but it's the thing. Don't want to give it away. It's the thing that every living thing needs these things. Every living thing needs these things to exist. Okay. Typically, there it needs to be there. All right. Some of those, hopefully, you can eliminate right off the bat. Okay, timer's on. Right there. Okay. So it would have to be able to use all the nutrients listed there. Okay. Okay. Let's see what we got. Okay, so we kind of split between B and C. Okay. What was that total? So that's an 80%. Okay. Between C and D. Okay, so who picked D? D as in dog, sorry. D as in dog. Anybody? Who? You? You picked D? Who picked D? Anybody? Somebody picked D. Majority picked D. So I know somebody's, you can't be that shy. D as in dog. Who picked D as in dog? 40-something% picked D. Did I say C and D? Oh, I said B and C. Let's see. That's entirely possible. Oh, you're right. B and C. Okay. Yeah. Okay. B and C. Okay. So the criteria was be able to use all of those. Okay. So a lithotroph can lithotroph use glucose? No. No. So if I left glucose out, I would be in a question like this. I'd be very specific, right? I would leave glucose out, put CO2 there or something. Lead you to if it were lithotroph, that's what it would be. Okay, so it's not that. It's not going to be anything autotrophic because there'd have to be some indication of that this is present, is there. Okay, so we have to eliminate everything that's an autotroph. Okay, so let's look at these two. Okay, who picked D as in dog? Anybody? Why? Had to be a choice. Had to be a reason. Right? Is there something missing? Okay. Is there something? Nitrogen's missing. Okay? So there's no nitrogen. And there's no nitrogen in the medium formulation. Right? Nothing with an N or an NH4 something, NH3 something, right? Nothing indicating nitrogen is present in the medium, right? So you picked heterotroph, you were right, but not any heterotroph would grow. It would only be one of these, okay? Because there's no, so the thing I was trying to hint at the start of the question was the old CHONPS, right? And the N's missing here, okay? So nitrogen fixing type would grow on that. So it's also an example of that medium being in the slide with Winogradsky and all that mentioned enrichment culture, right? Of an enrichment culture, because we take, say, a sample of soil, right? There's going to be a lot of stuff that's going to grow. But we can enrich for a specific type, like, say, if we wanted to look for nitrogen-fixing heterotrophs, right, by simply taking out the N. It's going to be very restrictive because the types that can fix nitrogen, there's a number that can do it, but not, not a whole bunch. And so that's a way to enrich that medium for the growth of those types. So it's a technique used to kind of, if you're looking for a certain type of bacterial type, kind of you want the conditions so that you'll favor their growth, and that's what the enrichment is about. Okay. Any questions about that? Okay. So this is really more what I talk about in chapter 4. Okay. So we'll see this stuff. We'll see this stuff again. Yeah. Question. Right there in the middle. C is for H for hydrogen, O oxygen. Uh, yeah, you'll know exactly what that is by the end of the semester. CHONPS. Any other questions? Okay. Yeah. Oh, right in front of you. Good. Yeah.
So, I understand why it could be answered in the additive scope, but wouldn't some chemical compounds be toxic to other additive scopes?
Oh, sure. It can be. But
Not out of that.
not that's we're just restricting it to leaving out one of those essential atoms. But fortunately, but we know that there are organisms that can fix nitrogen, right? So we know that if they're there, we have a chance of getting them. Okay. But yeah, there can be some things you might add that could be toxic to a certain type. Sure. Mm-hmm. Any other questions? Okay. Yes. Yeah.
That's what the heterotrophs can use, right?
That's what every living thing needs those, because that's what makes up most of your biomolecules. So everybody's going to have those. Okay. Okay. So let's. Okay. So classifying. So classification of microbes poses a bit of a problem, at least it used to, because of the species definition. Eukaryotes have, for the most part, two sexes, right? Male and female. There's sexual reproduction occurring, and you can define species as a group that only breeds within that group, right? So humans won't mate with tigers, okay? Because, not compatible, right? Don't breed outside of the species, okay? So that's a convenient way to kind of define various species of animals, etc., right? But we do that with eukaryotes. They don't fit that definition because they divide by binary fission, okay? For the longest time, etc. Are they in chains? Are they in pairs, or what have you? And then, aside from that, is metabolic. We learned about metabolism. Thanks. Okay, well, it can eat this or eat that, make the end product, and so we can leave metabolism microscopy. And so, because of the microscopy part of it, the classification of microbes changed over 200 plus years because of improvements in the microscope. Okay, so initially, well, let me just say, if you were in a lab, you learned this last week. The concept of resolution of a microscope, right? So if you have good resolution, you can have 2 points very, very close, and you still see them as two distinct points, right? So that limit of resolution is how close they can be, and still see them as two distinct objects, right? You have that, you have good resolution, right? So the microscopes weren't great at that, okay? And so you basically had to rely on, poor resolution microscopes, and so when you do that, basically microbes, initially it was about microbes either fit in animal plants. This is like back in the 1700s, and Linnaeus, if you remember him, that all microbes were lumped into one group or the other. I assume if they were greenish looking, it's like, oh, it looks like a plant, let's put it in this kingdom, okay? If not, then it goes into animal. So it was that way for a couple hundred years, well, at least 100 years plus, until we then took all the microbes out and put them in Monera. Okay, so prokaryotic microbes are in that group. Okay, so microorganisms are now in their own kingdom Monera. Then we had the improvement, basically the development of the electron microscope occurred. And now we could see the ultrastructure of cells, right? We could see organelles inside a eukaryotic cell, for example, right? We could see a nucleus in a eukaryotic cell. And so that's where we then get separation of the bacteria, prokaryotic types, okay, are in one camp, and then your eukaryotes in another. So that becomes the protista, okay, algae, protozoan. So clearly, unique cells with a nucleus and organelles and all the features of prokaryotic cells, okay, lacking in the prokaryotic types, okay. And basically all the prokaryotes are also just called bacteria, okay. So this would have been like 1930-ish or so, okay. Problem. They had been in plants for the longest times, but then somebody realized that fungi are actually more closely related than plants, because fungi are heterotrophs. They basically secrete enzymes that digest stuff out and they bring it in, like an absorptive type of digestion. But among the fungi, yeast, you can kind of out and whatnot. Okay, so once this was established, this stayed a fungi, so we have animal fungi, protista, this group of bacteria, and then we come around to domains, right? 1970s when this occurs. So the problem here is that among in trying to look at all life, okay, and to come up with a unifying way to compare all forms of life, let's use a molecule that would have been around, right, for a long time, would not have changed a lot, right, would have been kind of rose. And if you may or may not have known this, but in our biochem department, Dr. Fox, that's that Dr. Fox, George Fox, okay. Both of them came up with this way to use this use the ribosome. The ribosome, actually the gene, the DNA, the gene that codes for a particular part of the ribosome, right? It's called the 16S. And so S is simply just a unit of mass, right? You have a big S number, you have a bigger molecule, basically. Okay, that's all you need to know. Okay, so looking back, prokaryote ribosomes, okay, they have this structure. Okay, so basically a ribosome is made up of 2 units, and an RNA is synthesized, let's say, okay, and it combines with a bunch of proteins. Okay, and that's what makes up a ribosome in the most basic sense, right? A ribosome, and so they come together, fold up to form the ribosomal subunits. Okay, so we differentiate based on size, which we use with this S unit. Okay, so the small subunit is a 16S RNA, ribosomal RNA. Large one's a combination of 5S and 23S, so a super tiny one and a large one, and a bunch of proteins. Okay. And so they come together to form the 70S functional translating ribosome. Okay? So in eukaryotes, the equivalent molecule to the 16S is the 18S. Makes up the small subunit, plus a bunch of proteins. There's actually 3 ribosomal RNAs that make up the large unit. Okay? So the point here is that the ribosome is universal. Okay? It's super critical. Okay? If you can't make proteins, you will not survive. You just can't. Okay? So it's going to be a molecule that would have changed kind of slow. Okay? So things evolve at different rates, right? depending on the pressure. But for certain molecules, they change so much. Okay, you can't acquire any changes because you're going to lose function. Lose function completely if you do too many changes. And with the ribosome, you see that there are some changes, okay, over long periods of time. But these changes kind of we found with this technique that there are actually, within prokaryotes, two distinct types of prokaryotes. That was kind of the big thing about this was, okay, we have this group, but we think we're all the same. We're just bacteria, okay? And here's, if you laid out the ribosomal RNA, it would look like this, okay? And they have little loops here, areas of homology. The arrows are kind of areas where changes occur, okay? The other parts where you don't see it really doesn't change, okay? And so the archaea look like that, okay? And so this is the group that was found. So a very distinct structure, still a 16S, but just a different shape to it, slightly different shape and structure to it, that all of these possessed that were not possessed by bacteria. So different structure and shape to it, right? So enough changes that's significant. That identifies them as a unique group of prokaryotes, okay? And so it turns out that many of these members in this group tend to live on the extremes on Earth, extreme conditions, okay? Extremophiles, we call them, okay? And, of course, here for references, the 18S, ribosomal RNA. So with this technique, and you could make life as you could be in this group, they have the 18S, eukarya. If they have this form of 16S, archaea, this one, bacteria. So now we have two groups of prokaryotes. Didn't think that was even the case, okay? And so the looking at the 16S ribosomal genes, so remember that not all genes code for proteins. For some, the end product is the RNA. And that's what this is. Okay? So we can, so taxonomically, it's really examination of this gene for this ribosomal RNA. It's how we identify all things nowadays anyway, right? And we use it in addition to microscopy and metabolism, metabolic features, but certainly this is always part of the equation in doing identifications typically, okay? And so if we look at domains, so domains is a huge group, okay? It's the most inclusive group, the high, right? Phylum, class, order, blah, 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 blah. Okay. So domain's the biggest, so those. Bacteria, archaea, eukarya. And then kind of the phylogenetic tree, and then similarities. So archaea have similarities to bacteria and eukaryotes, okay? They lack a nucleus and no organelles. So these are hallmarks of any prokaryote cell, okay? And archaea are definitely prokaryotes because of that, okay? Also gene structure, metabolism. Now there's the eukaryotes. In some of their molecules, translation, polymerase, for example, similar most to eukaryotes. Also, you may or may not recall the gene structure of us and other eukaryotes is like the intron-exon configuration. Archaeal genes, many of them have that structure to them. So they do have that kind of eukaryote features shown here, okay? So, so just, I'm going to, oh yeah, that's what I want to do. So just real quick, kind of some of the features of prokaryotes. I think we're kind of know the basics of it, right? Like no organelles, asexual reproduction, right? Among the archaea, right, we lump them into three groups, right? Thermophiles, antigens, and halo. Yeah, can produce methane. Halophiles, salt conditions, hyperthermal. Then among eukaryotes, fungi, protists, photosynthesis and heterotrophy, and okay. So again, their own shelf. Viruses aren't cells, but they are microbes. Kind of a gray area, but they're on their own unique thing. Okay, so that's a good place to stop, folks. We'll see you. Well, we'll see you until Monday. So have a good weekend. Yes. Yes.
Hi.
Hi.
My name is Mir Khan.
Yeah.
I emailed you before the break started. I missed the first week of school.
Okay.
And is there anything that I missed that I can make up for?
No. All you got to do is, so everything you see and hear, see on the screen and hear, those are all captured. So just go into Blackboard. There'll be a folder called Video Lectures. So you'll be able to see what you missed last week.
Okay, but there's no assignments or anything.
There's the homework. That's part of the grade. So you still have time to catch up on that. I think. But do you have the homework?
Oh, we're talking about the chapter 1 of 3 homework.
Yeah.
Oh, yeah, I took note of that.
Smart work.
Yeah.
Yeah. Okay. So that's the only thing that's coming up immediately you have to do.
Okay. Otherwise, you should be good. Okay, thank you.
Hello, my name is Anika.
Hi.
I'm
What's
gonna
up?
actually